Hi, everyone. My name is Charlie Childs, and I recently defended my PhD at the University of Michigan in Ann Arbor. I just wanted to start by thanking everyone at Equalize for this amazing opportunity and all of you for attending today's pitch event. I also wanted to give a super special thank you to my fabulous mentor, Carol Curtis, who has given me invaluable guidance and support throughout the program. So I'm really excited to tell you guys about the company, Intero Biosystems. We recently spun out of the University of Michigan, in no small part thanks to this program, and our product GastroScreen, which we believe will revolutionize the way drugs go to market. So Layla did a really great job of introducing this concept earlier, but I just wanted to remind everybody of the problem at hand. The current drug development pipeline requires pharmaceutical companies to make a massive investment when they're looking for their next product. They start by screening hundreds of thousands of compounds for the potential to both treat disease and have a low amount of excess off-target effects. Promising candidates will then proceed to human clinical trials, which may be some patients' last hope. If a drug proves to be safe and effective, it will be approved by the FDA and go to market. To get a single drug to market, companies will spend close to 15 years and billions of dollars per compound that enters clinical trials. The cost of getting one drug to market is estimated to be so exorbitantly high because it factors in all the candidates that fail to reach market. In fact, 90% of drugs that go into clinical trials will fail. And most drugs fail because of unexpected side effects that were not predicted during preclinical testing. So how can such a long, regulated, and rigorous pipeline have such a high failure rate? It's because the current standard models used in the preclinical space are not able to accurately predict how drugs will behave in humans. So there are currently two different standards. The first are cell lines, and these are commonly derived from cancer cells and are far more simple than a human organ. Not only do they not accurately mimic the complexities of the human body, they are not even a model of healthy human cells. The second model are laboratory animals, and beyond the ethical issues associated with animal testing, humans are not mice, cats, dogs, or monkeys, and can react completely differently to drugs. These models cannot predict side effects in humans, leading to unforeseen side effects in clinical trials and resulting in fewer life-saving drugs reaching patients in need. So to address this challenge, major regulatory agencies across the globe are passing legislation to allow preclinical testing to be done without the use of animals and instead in more predictive human models. Organoids have been named as a possible alternative, and organoids are just miniature models of human organs that are designed to mimic the organ structure and function in a dish. Though small in size, organoids are highly complex and can be produced at scale, making them an ideal tool to predict how drugs will behave in a human organ. So to address this growing need for more predictive preclinical models, our company has developed GastroScreen, a mini human intestine that can replace the pre-existing preclinical testing models. Our patent-pending technology is the first and only stem cell-derived human intestinal organoid that captures the function and complexity of the human intestine. It contains functioning neurons, blood vessels, muscle, and the inner lining of the intestine, which comes in direct contact with any drug taken by mouth. Because of the presence of these cell types, which are spatially organized just like you would see in the real organ, it's possible to not only model healthy intestines, but also complex diseases. GastroScreen also has applications outside of the GI tract, as oral medications are absorbed in the intestine and allow for the potential of adverse side effects to be predicted in our product. Think of chemotherapies or GLP-1 agonists that have bad GI symptoms. GastroScreen can be used to test drugs in human organs before going to human clinical trials, mitigating risk, reducing cost in clinical trials, and identifying more promising compounds for patients in need. So here I'm showing you 3D images of the current state of stem cell-derived human intestinal organoids on the left, and a beautiful image of GastroScreen on the right. And what I hope you can appreciate is the vast increase in complexity GastroScreen offers compared to the status quo. In green, we have bands of smooth muscle that can spontaneously and rhythmically contract. In blue, we have neuronal networks that can respond to stimulus. And in pink, we have blood vessels that can actually have liquid flowing through them. In fact, compared to all other commonly used or commercially available models of the human intestine, GastroScreen is far superior and more complex because it is made of human cells, contains the major cell types of the intestine, is 3D with proper spatial organization, and it can mimic the functions of the human intestine, making it the most faithful model of the organ currently available. It can also be sold at a reasonable price compared to other preclinical models. Our team is comprised of leaders in the intestinal organoid field that will help ensure our company's success. I optimized GastroScreen throughout my thesis research by defining the first stem cell-derived human intestinal organoid protocol our faculty mentor at the University of Michigan and CSO Jason Spence first invented in 2011. Madeline is a biomedical engineer with experience in cell manufacturing in the industry space and will help to ensure that we can make these organoids at scale when it's time to go to market. So let me tell you a little bit about the business. GastroScreen will enable researchers from both academia and industry to test drug candidates in a human-specific context, allowing them to fail faster, reduce the use of animals, and improve the potential of their results to translate to humans, all while saving them money. We've assessed the market, and we know that there is a large opportunity for our product. Even with 5% penetration of our SOM, which comprises of the ADME toxicity and drug discovery markets in North America and Europe, we would generate close to $1 billion in market share. There are currently no companies making stem cell-derived human intestinal organoids. Our main competitors in the space are companies selling 2D monolayers, such as Altus and Maddox, organ-on-a-chip companies, such as Emulate and Mimetas, and companies that sell patient-derived organoids made of one single cell type, like Milliporcigma and the Hub. Our product is the only one available that's made from human cells, comes from the highly tractable and scalable source of human stem cells, contains the major cell types of the human intestine, spatially organized and in 3D. GastroScreen's ability to mimic the functions of the intestine is something that really differentiates our product from our competitors, as it has the ability to mimic major overall functions of the organ, such as the rhythmic movement of material through the intestine, known as peristalsis. We've spoken to over 50 stakeholders in the field, including 34 scientists and industry representatives, and during these conversations, 3 major pain points were identified. These included that current models were not complex enough, animal models were too expensive, and that Matrigel or other 3D matrices made the adoption of 3D cultures difficult and hard to scale. In fact, 100% of these stakeholders identified at least one of the following pain points. GastroScreen addresses the first 2 pain points, and we have ongoing R&D efforts to address the final. Of those we interviewed, the majority of them were actually already using some sort of simple 3D model in their research, but expressed interest in a more complex 3D model. The next major category were those that were not currently using, but were very interested in using complex 3D models, with a common theme being that upper management was pushing for them to integrate these models quickly into their workflow. And finally, a very small portion were interested in 2D models only, as they had already heavily invested in the system. Of those interested in using complex 3D models, there was a relatively equal interest in use cases, including viability, cytotoxicity assays, disease modeling, and transcriptomic analysis, all things which GastroScreen can do. Based on these efforts, we plan to market GastroScreen in 2 different verticals. The first is a model of the human intestine in both a healthy and diseased state, and the second as a screening platform which could be used for all oral drugs to test for adverse side effects. The first vertical will have organoids that can be bought by the plate, with a target customer of researchers interested in using organoids in their internal research programs. This would feature straightforward pricing, where 96 well plates with 1 organoid per well will be sold for between $5,000 and $10,000 a plate. We are still actively collecting pricing data, but we believe this is a pretty solid range based off of our discovery so far. The second vertical would be a fee-for-service model that other companies in the space have seen great success with. The target customer are drug companies that may not have the expertise or equipment in-house to grow these organoids, so we would run all the preclinical assays for them and provide the data. While pricing for these services would be more customizable and on a case-to-case basis, based off our competitors in the field, we believe we can start pricing at around $30,000. To date, we've raised over $100,000 in academic commercialization grants and have applied for an STTR, which will support our planned lab work, including benchmarking GastroScreen's response to drugs against other preclinical models and human data. We are actively developing other products, such as disease models, more complex preclinical tests, and organoids of other human organ systems, which we are actively seeking grant funding to execute. We plan to run pilot testing in early 2025, launch a commercial product shortly after, and we anticipate raising a seed round by the end of 2025, as we are currently evaluating capital access strategies. We are still early to consider exits, but we do believe that an acquisition by a large pharmaceutical company, a CRO, or other complex in vitro model companies would be a potential exit for our company. We know the value of talking to our customers early and often, and with all the great connections that all of you have in this room, we would ask your help in making connections to potential customers and key stakeholders as we continue to develop this product so that it is well positioned when we're ready to go to market. So we believe the future of preclinical testing is miniature organs, and that GastroScreen and Entero Biosystems will lead the charge to help identify safer, more effective drugs for patients. My contact information on the screen. Please feel free to scan the QR code to collect it on your phone, and I'm happy to take any questions. Thank you.